introduce Pam Soltis. It's really exciting to have her. She's calling in from, zooming in from Florida. She just zoomed in from Florida. And I'll just introduce her briefly so you get to know her a little bit better. She's a distinguished professor and curator in the Florida Museum of Natural History at the University of Florida and serves as director of the UF Biodiversity Institute. Her main research interests are angiosperm phylogeny, polyploidy, and the use of digitized natural history collections in biodiversity research. Working with Doug Soltis, her husband, and others, she has used a phylogenetic framework to address patterns of genome evolution and ancient polyploidy, including ancient whole genome duplications that mark key clades of angiosperms. She has also been active in scientific outreach that combines science and art, focusing on understanding, interpreting, and using the tree of life. She served as president of the Botanical Society of America, of the Society of Systematic Biologists. She is currently past president of the American Society of Plant Taxonomists and the president of the International Society of Phylogenetic Nomenclature. She was recently elected to the U.S. National Academy of Sciences, and she's also a fellow of the American Academy of Arts and Sciences. So with that, we'll let Pam take over. Welcome, Pam.
Great, thanks a lot. Thanks so much for the invitation, and I'm looking forward to having a chance to chat with you following my presentation as well. So I'm going to be focusing today on polyploidy as a process and polyploidy as evolutionary patterns. And I like to think of polyploidy as a process that integrates from cells to ecosystems. So the sub-theme here will be integration from cells to ecosystems. And I'll start off right now by saying that all of the work that I'll be describing today was done in collaboration with Doug Soltis. And here he is very fittingly standing in a field of polyploids in the Alps when we were visiting Zurich a couple of years ago. So there are a lot of cardamine polyploids around his feet in this photograph. Now, plants are a major component of the tree of life, with potentially close to 500,000 species of green plants. And these green plants trace their evolutionary history back approximately 1 billion years, and they represent spectacular diversity in many aspects of life. But one of the things that I think is most remarkable about the green plant clade is the immense diversity of genome sizes, with genomes that range in size from 50 megabases to 150 gigabases. Yes, megabases to gigabases, and this represents about a 3,000-fold difference in genome size. So how did this variation in genome size arise? Well, there are two main processes that we think contributed to this variation. One of these was transposable elements. Certain groups, such as gymnosperms, have amazing proliferation of certain classes of transposable elements and various repeated elements. And this is responsible for a lot of the variation, perhaps most of the variation that actually exists in the sizes of the genomes. But I think it's polyploidy, or whole genome duplication, that really contributes to the complexity of plant genomes and also to much of the variation in size. And so just for reference, there are some plants that have a haploid chromosome number of 2, so that their diploid sporphytic number is 4. But there are others, such as this fern, Ophioglossum reticulatum, which has 630 pairs of chromosomes in the sporphytic tissues. So that's quite an immense number. And it, of course, raises all sorts of questions about how these chromosomes find each other, even in for meiosis, how they go through these processes, how they have the energetics to continue to support a genome with this sort of size and complexity and so on. So polyploidy has been recognized as a prominent feature in plants for a very long time, close to a century, in fact, and about half of all flowering plants are considered to be of polyploid origin. That, of course, is our traditional estimate when we go to all of the classics, Stebbins, Grant, and so on. But stay tuned, we'll have an update on that perspective in a few minutes. It's also estimated that about 95% of all fern species may be polyploid, and most of our major crops are polyploid. And these include these examples that I've listed here: wheat, corn, sugarcane, cotton, potato, coffee, the brassica crops, and more. In addition, polyploidy often confers very good colonizing ability, and 17 of the 18 world's worst weeds are also polyploid. Hundreds of ancient whole genome duplications have been identified across the plant branch of the tree of life, and some of these ancient whole genome duplications map to the timing of major geological events, such as the KPG or KT boundary, and much of this work has been conducted by Yves van de Peer, who has really enlightened us about these associations between major geological events and the presence of whole genome duplications. So I think you can see just from this summary that polyploidy has tremendous evolutionary, ecological, and economic importance. And the more we can learn about it, the more we can understand plant evolution, the more we can understand the adaptations of many plants to their environment. We might be able to use this information to help us predict how plants will respond to climate change. And of course, the tremendous economic importance associated with so many crop plants is also evident. Now, the important role of gene duplications in evolution was first really noted by Susumu Ono, who in 1970 published this book, Evolution by Gene Duplication. And in his work, he acknowledged that perhaps the most important source of genetic variation is duplicate genes, because these duplicates can diverge from each other. One copy can accumulate and tolerate mutations while the other copy maintains the original function. And this provides any set of genes, any pair of genes that's been duplicated, whether by polyploidy or some other mechanism, to generate genetic variation that could result in novelty. Now, of course, when we're considering polyploidy, when the whole genome duplicates, that provides an immense and immediate source of genetic variation. Now, in 1983, Don Levin published what I consider to be one of the most influential papers in the history of polyploidy research. And this is entitled Polyploidy and Novelty in Flowering Plants. And in this paper, Levin considered all of the downstream consequences that a change in cell size due to a duplicated genome might have. And these so-called nucleotypic effects have effects at the cellular level, the biochemical level, physiological, developmental, ecological, and ultimately evolutionary scales. And so the result of all of this is that polyploidy can generate immense novelty, essentially immediately following the production of a polyploid lineage or species. More recently, Jeff Doyle and Jeremy Coate have followed up on this theme of nucleotypic effects in this culture review that was published in 2019 in the International Journal of Plant Sciences. And this is an amazingly comprehensive review of these same sorts of concepts that Levin presented, but with an updated perspective and a lot of references as well. So I would direct your attention to this paper as well if you're interested in some of these nucleotypic effects. But there are many other effects of whole genome duplication. These include gene family diversity, processes of subfunctionalization and neofunctionalization. There are new combinations of genes that can be developed through polyploidy polyploidy. And the result of all of these processes is the potential for evolutionary and genetic novelty. I think we all recognize polyploidy as an important speciation mechanism. The classic work of Clausen, Keck, and Heisey really points to specific examples, but also plays up the importance of this speciation mechanism. But polyploidy is more than a speciation mechanism. As I've mentioned, it generates genetic and phenotypic novelty, and it shapes macroevolutionary diversification. And because of these different effects at these different scales, from the microevolutionary to the macroevolutionary, I really like to think of polyploidy as this integrator across levels of biological organization. Now, in my presentation today, I'm going to focus largely on the work that we've been doing on polyploids in the genus Tragopogon over the past few decades. And I'll use this as a jumping off point to investigate some additional themes about the origins of evolutionary novelty in polyploids in general. So in this particular system, which was described by Marion Ownby in 1950, three diploid species of Tragopogon from the sunflower family were introduced from Europe into the Pacific Northwest of the United States in the early 1900s. And these species were Tragopogon pratensis, Tragopogon dubius, and Tragopogon porifolius here at the points of this triangle. And these diploids hybridized with each other and produced allopolyploids. So Tragopogon dubius and pratensis formed Tragopogon misellus, and dubius and porifolius formed Tragopogon mirus. So this shows the relative geographic scale for these species in this, the Pacific Northwest region of the US, and primarily in this very narrow area here in this blue box in eastern Washington state and western Idaho. And these polyploids were formed sometime within the last 100 years. We think probably within the last 90 years or so, because the diploid species had not been introduced into this region until the mid-1920s. Now, these polyploid events then are somewhere around 40 to 45 generations old, because the plants are typically biennials. Now, the polyploids and the diploids as well occur in weedy sites, such as along roadsides or railroad tracks. And this shows a classic habitat here, actually between a highway and the railroad tracks. Now, in some of our earlier work, we were interested in the concept of multiple origins of these Tragopogon polyploids. And in fact, this concept in Tragopogon goes back to the work of Ownby himself, who identified separate individual populations of the polyploids that differed morphologically and chromosomally. But over the years, various sorts of biochemical and molecular techniques have been applied to investigate the possibility of multiple origins of these polyploids. And we now recognize that just about every population of each polyploid species probably originated independently from localized diploid parents. So just to illustrate this, these are structure plots based on microsatellite data for Tragopogon mirus on the top and Tragopogon misellus on the bottom. And each column represents a single individual, and each color block represents a separate population. Now, the reasons that these are in color blocks like this is because all of the individuals are identical within each of those populations for the microsatellite markers that we used. So we have populations from throughout this narrow geographic range in eastern Washington and adjacent Idaho, where every population is genetically uniform, but all of the populations differ from all others. With the single exception of this one individual way on the right that has, let's have a different color, and it actually represents the product of gene flow from one population to another very nearby population. So that's in Tragopogon mirus, and then in the bottom, Tragopogon missalis, a very similar pattern, although there's a little bit more genetic diversity within some of these populations. But essentially, every time almost every time the diploid parents get together, they form a polyploid in that location. So we have a series of natural evolutionary replicates that allow us to investigate what happens following polyploidization, not after a single event, but after multiple events, all in a natural environment. So for about the last 35 years or so, Doug and I have been studying these polyploids, and we've been interested in the extent of additivity of the parental characteristics that is found in each of the polyploids, and also in the amount of novelty that we see in the polyploids relative to the diploid parents. And we've been studying this at both the genetic level and at the chromosomal level in both natural allotetraploids and in synthetic allotetraploids that have been generated through our research program. And again, the multiple origins of each of the polyploid species provide us with natural replicates so that we can evaluate the possible common aspects to polyploidization and the common impacts that polyploidization may have. So before describing some of this work, I'd like to make sure we're all on the same page with regard to a little terminology. And so I'll introduce the term homeolog. And homeolog has been used to refer to duplicate chromosomes or genes that are derived from the different parental species in an allopolyploid. So here we see the chromosomal complements of two diploid species that are then united in an allopolyploid species. And we have this chromosome pair and this chromosome pair, which seem to represent related chromosomes in these parental species. And then when they're united in the allopolyploid, they become referred to as homeologs. And this is in contrast to, of course, the two chromosomes of the pair. Those are homologs. These are homeologs. And here's what this would look like when we actually look at the chromosomes of one of the Tragopogon tetraploids. Again, the duplicate chromosomes derived from the different parental species in an allopolyploid. And we have the component of the one parent here on the top in green and the other parent on the bottom in red. And so we've been asking this question of the Tragopogon allotetraploids. Are the allotetraploid Tragopogons perfectly additive of their diploid parents? And we've been investigating this question from the gene content, gene expression, and chromosome perspective. Because of course, if we think about the classic view of polyploidy, it would look something just like this figure, where we have a chromosome complement from one parent, the chromosome complement from the other parent, and everything is perfectly additive. But I think if you look a little carefully, even at this diagram, you can see that this chromosome pair here, chromosome pair C from the P genome, which is generally orange, has a little bit of green on the bottom. So there's some genetic material from the D genome that's been translocated there. So there may be additional duplications of genetic material in that respect. And there are other ways in which non-additivity may arise. And that's what I'm going to be focusing on for the next few minutes. So we were interested initially through some of our molecular studies in trying to determine whether all genes were actually present in duplicate copy, or whether there might be some variation in terms of some copies being lost from one parental genome or the other. And so in some work that we conducted several years ago with Pat Schnabel and Brad Barbazuk, along with former postdocs Richard Buggs and Ingrid Jordan Thaden and a sabbatical visitor, Leaderson Vicini, we investigated this idea of homeolog loss in the two allotetraploids. And we surveyed about 450 homeolog specific SNPs and looked for presence and dosage in the tetraploids. So essentially, we were interested in inferring whether or not there were two alleles from the dubious parent and two alleles from the porphyrious or pratensis parent in each individual. And then we scored the presence of all of those alleles or deviations from that purely additive pattern. So in this graph, we're just summarizing the amount of duplicate genes found in overall across a number of samples of Tragopogon mirus. And we find that from our natural population survey, 72% of the loci in all of the individuals sampled are present in duplicate, which means that 28% of them show loss of one parental gene copy or the other. And in red, we see the value for the synthetic polyploids. It's a little bit higher. There hasn't been as much loss. And this is only through the first 5 generations of synthetic polyploids. We see almost identical results in Tragopogon miscellus. Again, 72% of the loci are duplicated, so 28% were lost. Or show loss of one parental copy or the other. In this case, the pratensis and dubious losses are about equivalent, whereas in mirus, there were more cases of porphyrious parental loss than dubious loss. Now, we did actually investigate these patterns across the multiple origins, and we see quite similar results with the percentage of duplicates ranging from 65 to 78% of the loci surveyed, meaning that there was 22 to 35% loss. And in general, there was more loss of porphyrious than dubious across all of these populations. And again, quite similar results for the other tetraploid, Tragopogon miscellus, with 61 to 79% showing duplicates and 21 to 39% showing loss. And again, across all of these populations, on average, the dubious and pratensis losses were approximately equivalent. So we put this together in a large grid format, and I'll explain how this is set up here before going into any more detail. So across the top, we have the individuals surveyed, and those to the left represent natural populations, and those to the right, the synthetics. We're starting out here first with just Tragopogon mirus, and then the rest of this figure is set up actually as a grid, where we have the loci down the side, the individuals across the top, giving us this grid shape. And everywhere where there's black, it shows that the patterns are additive and that both parental homologs are present. But everywhere you see pink, that means that there's a loss of the porphyrious, either allele or both alleles. And where it's yellow, there's a loss of one or both dubious alleles. So you can see when you look at this figure, I think that most of the loci show the additivity patterns that I just described from those previous histograms. So 72% of the loci don't show any homolog loss, but we do see pretty evident loss of the porphyrious alleles, particularly up here. There's a whole set of genes that show loss of the porphyrious copy, and down here a set of loci that show the loss of the dubious copy. Now, unfortunately, because of the nature of the markers that we used, it's been difficult through this analysis to identify what these loci might actually represent. So we're really interested in digging into this a little bit further to find out what sorts of genes tend to be retained in duplicate and which ones tend to have the loss of one parental type or the other. And this shows the results for Tragopogon miscellus. Very similar sorts of patterns, with most of the loci being retained in duplicate, but big chunks that are routinely lost from one parental genome or the other across most individuals that were sampled. And you can see that up here, for example, at the top where we have the loss of these dubious copies across just about all of the individuals, or down here, the loss of the pretensis genes across just about all of the individuals. So when we ask whether or not the tetraploid species of Tragopogon are perfectly additive of their diploid parents with regard to gene presence or absence, the answer is no. And that when we look at any single individual, every individual is genetically different from every other individual in the patterns of duplicate gene retention and loss. So there's definitely novelty that's being generated through these patterns of homolog loss. We see very similar sorts of patterns when we look at homolog gene expression. So the previous analysis was done with genomic DNA. This was done with cDNA. The grids are set up in the same way. So we see, for the most part, duplicate gene expression. But in some cases, we see the pink bars, that's the silencing of the porphyrious parent. And in other places, yellow, silencing of the dubious parent. And the same thing is true for the Tragopogon misellus individuals, with more, in this case, silencing of dubious than the pretensis copy. You can see some pink here, but not nearly as much. We've also done some additional analyses of gene expression using transcriptome profiling and some specific sorts of transcriptome comparisons that I'll describe in a minute. Our initial work in this area was led by Lucas Boatwright, who was a PhD student working with Brad Barbazuk at the time, and Mi-Jung Yu, who was a former postdoc. And this work was published in Genetics a few years ago. And in this study, we examined transcriptomes of leaf tissue across multiple replicates of each of the 5 species. And in this analysis, we identified what we called common orthologous regions that were identified based on sequence similarity between the diploid parental sequences. And we defined this common orthologous region as a target then for looking for homolog specific expression in the tetraploid species. So in the first example, this is Tragopogon misellus first. We identified about 15,000 of these common orthologous regions. After doing some filtering to remove bias from the data set, we settled on about 7,000 loci for examination of homolog specific expression. And of those, we found that about half of them showed no homolog specific expression. In other words, the parental copies were expressed equivalently. But of the other half, interestingly, about half of them showed a preponderance of pretensis expression, and about half of them showed dubious expression bias. So that information is just summarized right here. Again, half of the loci analyzed showed no homolog specific expression. And of those that did, half showed a dubious bias, half showed a pretensis bias. And gene loss, which is just noted here, is much less than the expression bias. We found almost identical results for Tragopogon mirus. Again, about 15,000 loci or cores were identified. About 8,000 were analyzed. Of those, half showed no bias. And then maybe slightly more showed of the remainder showed a porphyrious bias than a dubious bias. But in general, those were quite similar. Now, one of the really interesting features of this Tragopogon system is that for Tragopogon misellus, the yellow flowered tetraploid, there have been reciprocal origins, and that the reciprocal origins have a striking phenotypic difference. So the long liguled form shown here has Tragopogon dubius as the maternal parent, and the short liguled form shown here has Tragopogon pretensis as the maternal parent. And so we were interested in identifying possible expression differences between those two forms. So we analyzed samples from the localities where these species occur naturally in Washington and Idaho, and we subjected them to RNA-seq analysis. And here we were interested in finding out if there was a bias in the homolog expression or between the, for each of the tetraploid forms, or whether there was no bias. And so what that would look like is if there was no bias, there would be equivalent mapping of the expression to the, of the expressed genes to the two parental copies. If there was a bias toward dubious, we'd have more dubious homologs, dubious represented in orange, and here in Macellus, the blue representing pretensis. And so this would be biased toward dubious, and then the reciprocal bias toward pretensis. And this is work that Sheng-Chen Shan did as part of his dissertation work with us. So this summarizes the results here. About 80% of the loci show show no bias, and about 10% or so, 9 or 10% show bias toward dubious. This is true of both forms, and both forms also show about 10 to 11% bias toward pretensis. I have no idea where that mark came from. So we have another analysis that we did to investigate whether the loci that are biased in one way in the long ligule are also biased in the short ligule. So here we compared the patterns between them, and we found that about 82% of the loci show the same homolog specific expression profiles between the two forms, meaning that a very small number of loci, relatively speaking, is associated with one or the other phenotype. And those values are shown here. So about 18% show different homolog specific expression profiles between the forms. We are also interested in whether differential expression in Macellus, or how it related to the patterns of the diploid parents. And these diagrams illustrate different comparisons of the parental types with the polyploid. So here the D is dubious, the P is pretensis. Those are the two diploid parents, and M represents Macellus. So here, in fact, no change in gene expression is found for 76 to 80-ish%. And then all of the remaining patterns represent just the remaining percentage. So only 1% or so show additivity, about 5 or 6% show expression dominance toward dubious, about the same percentage toward pretensis. There are patterns of transgressive downregulation and upregulation and so on. Oops. So so at the bottom here, this just summarizes these non-additive expression patterns. So the short liguled form has about 23% non-additive expression, the long liguled form about 16% non-additive expression. And then when we hone in on those loci where we can see differences between the long liguled and short liguled forms, there are 39 loci that have that are upregulated in the long liguled relative to the short liguled form, and 2 that are downregulated in the short liguled relative to, no, in the long liguled relative to the short liguled form. And when we do investigate what those loci might be doing, they tend to be associated with pectin and other aspects related to cell wall modification and things that enable growth. And this actually is an interesting finding because of the fact that these represent different sizes of ray flowers or ligules, as they've been referred to. And so the fact that they may be associated with different growth forms and different aspects of growth provide us with some targets, some potential candidates for investigating further. Okay. So these last few examples on these transcriptome dynamics of the inflorescence can all be summarized here. So basically, most of the loci have unbiased gene expression between these two forms, the long liguled and short liguled forms. Most of the genes show the same expression pattern between the forms. Most of the genes share expression patterns with their parents, and it's only 22% in the long liguled and 16% in the short liguled that show non-additive expression. And pectin and other cell wall components are more highly expressed in the long liguled form, and these loci are some potential candidates that could explain the morphological differences. So when we return to this question of whether the tetraploid species of Tragopogon are perfectly additive of their parents with respect to gene expression, the answer is convincingly no, and we see a lot of novelty in gene expression patterns. Now, the last example that I'd like to share has to do with karyotype variation. We were interested at first in whether or not some of the homeolog loss patterns that we were observing might be due to changes in the karyotype. So whether or not there were shifts in the type or amount of genetic or of chromosomal segments within the allopolyploids relative to the diploids. And this is work that Mike Chester did as a postdoc with us several years ago. And according to this model, again, we would expect if we had two diploids and the formation of a diploid hybrid and eventually an allotetraploid, that we would get these perfectly additive allotetraploid genomes and karyotypes. But is that what we see? Well, in fact, not really. Using chromosome painting techniques that Mike perfected on these Tragopogons, we investigated the possibility of aneuploidy and its components in Tragopogon miscellus and in Tragopogon mirus as well. Here is some original data from the Tragopogon miscellus analyses. So the expected chromosome number is 24. We see some individuals with 23, and we're able to, through the chromosome painting and karyotyping, determine that there's been a loss of this E chromosome from the pretensis genome, subgenome. And here there are 25 chromosomes, and this represents an addition of chromosome C, also in the pretensis subgenome. And we found a fair number of these numerical aneuploids, but relatively small, maybe what one might expect. But what was much more amazing through the course of this work was the high presence of what's been referred to as compensated aneuploids. And these are aneuploids that have the expected chromosome number. So these all have 24 chromosomes, but they don't have half from one parental genome and the other from other half from the other parental genome. So for example, in this case, we see trisomy for the dubious set E, monosomy for the pretensis set E. Here, monosomy for dubious, trisomy for pretensis. And here, tetrasomy and nullosomy. So we found amazing patterns like this that were very common throughout the individuals that we sampled. And in addition to that, we also found evidence for translocations. So when we summarize the data, in fact, for Tragopogon miscellus, only 31% of the individuals had additive karyotypes, 10% were numerical aneuploids, but 59% were these compensated aneuploids. And these are a type of polyploid that that couldn't be detected with traditional chromosome squashes. So despite the fact that Ownby was amazingly successful at finding polyploids and documenting their existence, he didn't have the tools then to be able to determine whether something was an additive polyploid or a compensated aneuploid, such as most of these turn out to be. So when we look at Tragopogon mirus, though, on the other hand, we also see compensated aneuploidy, but in very different percentages than in miscellus. So with regard to the karyotypes, again, the Tragopogon tetraploids are not additive of their parents, and we see evidence for novelty again among many individuals. Now we're currently developing more genomic resources for Tragopogon. We have a draft genome of Tragopogon dubius. This was work done by Xiaoshan Liu as part of her dissertation a few years ago. And we're now working with Dovetail to produce a more contiguous genome for Tragopogon dubius. And more recently, Shengchen Shan has also developed CRISPR methodologies for working with Tragopogon. And you can see here his successful gene editing of the phytoene desaturase gene, making this an albino. And this was work that was done in collaboration with Bing Yang, formerly at Iowa State and now at the University of Missouri. One of the, in fact, bigger challenges, though, with the concept of gene editing and modification has been with the regenerating the plants. And Shan has recently been able to generate both shoots and roots from leaf explants. And this is true for both the diploid Tragopogon portfolius and the tetraploid Tragopogon mirus. So we're now ready to be able to investigate the effects of some of these loci that are held in duplicate or others that have shown a loss of one parental copy or another. So by doing some gene editing, we hope to be able to identify phenotypes that result from shifts in these patterns. Now we've also noticed that, well, we asked the question of whether this variation, this novelty we see in the Tragopogons of very recent age, these ones from the Pacific Northwest of the US, is that all just noise, or does that sort of variation maybe have some longer term impact on the evolutionary process? And we see, in fact, by looking at other polyploid complexes in Tragopogon, that very similar patterns exist. So on the left, we see the Tragopogons from the US, and on the right, a complex that occurs in Spain. And this has the tetraploid as Tragopogon castellanis and with Tragopogon lamardii and crucifolius as the parents. And this is work that Mike Chester and Evgeny Mavrodiev led. There are three origins of Tragopogon castellanis in Spain, marked by these three colors. The origins 1 and 2 have additive karyotypes, perfectly additive of the parents, just like what you might expect had you not seen the first half of my talk. But the third origin, in fact, shows patterns exactly like what we see in the North American polyploids, where there's compensated aneuploidy. There are 4 copies of the E chromosome pair from crucifolius and none from lamardii, and there are also patterns of translocations among the different chromosomes. And so we see some similarities to the younger species, compensated aneuploidy and translocations, as I just showed, but we also see more extensive evidence of homolog losses than in the recent polyploids. So it looks like these same processes that we see in Tragopogon mirus and Macellus can operate in other older polyploids and generate even more novelty over the course of evolutionary time. So I think what this shows us is that polyploids are mosaics of additive, parental, and novel traits. We see variation among individuals, and this variation may generate arrays of novel combinations. We see variation at the genetic, chromosomal, cellular, physiological, and ecological levels, as well as morphological levels. And I'm showing you data to back that up, but you'll have to believe me on that one. So polyploids are not composed of identical individuals. And so that leads us to ask what the evolutionary consequences of novelty might be. So all of this work that I've been describing is essentially variation that occurs at the population level. But over time, we go from this continuum of population level variation to species level variation to variation that is represented much deeper in phylogeny. And so this leads us to ask about the deeper level aspects of polyploidy. Now, we all have learned that about half of all angiosperm species are of polyploid origin. But in fact, when we do genome comparisons, we see that there is a whole genome duplication that marks all of the angiosperms. And this is this peak right here. So all angiosperms, in fact, are polyploid. And there's a deeper duplication as well that characterizes all living seed plants. Now, when we start to map out whole genome duplications across angiosperm phylogeny, we see some interesting patterns. So every one of these green starbursts represents a whole genome duplication. And in fact, basically every genome that's been sequenced has evidence for one or more ancient duplications. But what we see in particular are that these key points in flowering plant evolution, like the origin of the flowering plants themselves, the origin of the eudicot clade, the origin of the grasses, and so on. Many of our most iconic splits or divergences in the angiosperms are accompanied by ancient duplications. And this makes us ask the question about whether or not there's a relationship between whole genome duplication and diversification. Another example from some of our recent work occurs in the mint family, where we also see very extensive patterns of whole genome duplications, possibly 15 to 30 whole genome duplications across the history of the mint family, a family that's made up of about 7,000 species. So are whole genome duplications associated with major diversification events? Well, in order to address this question, we worked with some statisticians and some other phylogeneticists to ask if there was perfect correspondence between ancient polyploidization and increased net diversification, or whether perhaps there was a delay or lag in the elevated net diversification rates following polyploidization. And the reason we asked about this lag is because several years ago, Eric Schranz and colleagues proposed this concept of a delay or lag following in diversification following polyploid formation. And that was based on work from Brassicales. And we were curious as to whether the same thing showed up across the angiosperms as a whole. So we placed 9 whole genome duplications, and this was only a few years ago. And yet at that point in time, our understanding of both whole genome duplications and certain aspects of phylogeny were both had a lot of uncertainty. So we were only able to place 9 of these. These are shown with the stars. And in fact, we examined some alternative placements of a few of them. And then we looked for correspondence between increased or decreased rates of diversification at those same nodes. And only one of those duplications showed a perfect correspondence. And that was this one you can see here in the bottom. And this is associated with the origin of the Asteraceae. And so we see a significant rate of increase in diversification at the, associated with the Asteraceae and a whole genome duplication that corresponds to its common ancestor. So this is interesting because this isn't this is a clade that has undergone a lot of whole genome duplication. So we found that diversification shifts were rarely perfectly associated with duplications, and that the shifts typically did follow a lag. So this suggested that the whole genome duplication might be enough to promote, but not enough to cause increased diversification. And in fact, the data set was so small there, we started looking for more evidence so that we could test this more appropriately. So following just a few years, there were so many more genome duplications identified based on the work of the 1KP consortium that our former student, Jacob Landis, was able to do a much larger analysis of this question. So we used a phylogeny of about 30,000 angiosperms that had been published previously, and we were able to plot about 100 whole genome duplications thanks to the work of Mike Barker and his lab, particularly Zhang Li. And we identified a number of shifts in diversification rate. And we found, though, that only about 40% of these whole genome duplications were actually associated with upshifts in diversification. But about 2/3 were associated with increased species richness in sister clade comparisons. So this tells us that in many cases, these whole genome duplications were associated with upshifts, but this was not statistically significant. However, this very interesting point of having much higher species richness in the clade marked by the duplication versus its sister clade that didn't have it suggests that there may really be something going on here. So in answer to this question, are whole genome duplications associated with major diversification events? Well, sometimes. So again, about 40% are statistically associated with upshifts, and about 2/3 are associated with higher species richness in these sister clade comparisons. So this brings me to my final point, and this gets back to this concept of novelty and the role of key innovations in angiosperm diversification. Now, we tend to think of key innovations as being those characters that help spur on diversification. But what if those key innovations are actually the product of these whole genome duplications? So we're interested in looking at what those key innovations might be at some of these same clades where we see whole genome duplications. So for example, at the early in angiosperm evolution, we see the origin of vessel elements. We see the closure of the carpel, some of these major events that arose a few nodes past the actual polyploidization event. In addition, we see at the origin of the eudicots, the sort of the canalization of the pentamerous flower once we get a little bit inside the origin of the eudicots, along with these at the core eudicots, along with some chemical changes. And then, of course, many, many changes associated with monocots. So thinking about these concepts of novelty and diversification and what we've learned from our studies of Tragopogon at the microevolutionary scale, we've put together this cartoon of how we think novelty and diversification may come in line. So polyploid formation leads to lots of diversity on lots of sorts of scales. We see variation in morphology and gene expression and karyotype and gene content. And all of that diversity then is present in early stages following polyploidization. But then through environmental filtering, some forms of selection, possibly even genetic drift, one, let's say, or a few forms emerge. And following the right set of environmental conditions, that form could undergo rapid radiation. And this rapid radiation at a shallow point in time could end up having manifestations deeper in phylogeny, showing ancient radiations such as we see for the composite as a whole. So finally, we just are thinking about polyploidy or whole genome duplication as this major biological force that extends from cells to ecosystems. And this is from a recent review that we published with Don Fox, Thielen Ashman, and Eve Vandepeer in Trends in Cell Biology just last summer. And here we've just focused on how polyploidy has repercussions for biodiversity, biocomplexity, agriculture, biotechnology, and medicine. So to summarize, from our work in Tragopogon, we've shown that novelty arises quickly in polyploids. We see evidence for gene loss, homolog-specific expression, karyotype diversity, morphological variation, also some ecological variation, which I didn't share with you. And polyploidy may trigger diversification. Again, about 40% of the angiosperm whole genome duplications are associated with these upshifts, but that's not significant overall. But 2/3 of the duplications are associated with increased species richness relative to their sister clades. So we think that whole genome duplication may, in some cases, drive novelty, which could lead to key innovations. And this can lead to effects at the subcellular to ecosystem scales, with implications across many domains, from biodiversity to agriculture and medicine. And finally, lots of people contributed to the work that I've described today. And I hope I got everybody here from these main projects, but I tried to acknowledge the key people as I went along. So with that, I'd just like to say thank you to everyone for your attention, and I'd be happy to take some questions.
Hey, Pam, that was great. Thank you.
Thank you. Thanks.
Very exciting stuff. Very exciting. Do we have, I'd like to open up the floor to questions. Don't be shy. We ended up, we had 62 people. Well, actually, we had 66. So that was pretty good. Pretty good turnout.
That's great.
Yeah.
Oh, I mean, I could kick it off if you want.
Go for it, Mark.
I'd rather a student did, but at any rate, I've sort of got a pair of questions, if that's allowed. One is, it's interesting, this Tragopogon, you said, I paraphrase you, of course, my apologies, but you sort of said, anytime you got sort of two diploids together, they made a tetraploid. So I'm interested in the molecular, the molecular basis for that, because not every plant does that. And so just wondering if you are able to speculate or know the molecular basis for this propensity to form these tetraploids. And sort of the mirror image question, why are so many plants not polyploid in terms of, research? I know, I appreciate what you're talking about, that they're all polyploid. Yeah, well, okay. But in terms of recent polyploid, I'm thinking in particular about some of our personal research, our own research, where you've got the potato, which is this tetraploid, and then tomato, which is not a modern tetraploid, and wondering why some plants aren't like that when their near cousins are.
Yeah, great questions, and I think I think both of those are questions that that need a lot more attention. The question of why certain groups form polyploids and other groups don't is extremely interesting. I mean, we see within certain clades do it a lot, and other clades don't do it at all. So so is that ecological because they're separated, or is it genetic because maybe they make them, but there's they're immediately selected against, and so we don't ever see them. Really interesting question. And I think we really need to know a lot more about the whole formation process. So what triggers unreduced gamete formation? What allows those unreduced gametes to make a viable, a viable zygote, and then eventually, full fertile plant that can continue on. So just in general, I think we need a lot of work on that. With regard to the Tragopogons, it's a very interesting situation. So these three diploids occur in Europe. They don't occur sympatrically very often, but occasionally they do. And when they do, they almost never form polyploids in Europe. So, but there's one particular area in Sweden, one of the islands off of Stockholm, where the dubious and pretensus occur together, and there are hybrids and triploids, but no tetraploids. So there's kind of this idea that maybe something might happen there if people will just kind of keep an eye on it. There was a master's thesis from Stockholm University on that several years ago, where they had been following them. So, so why is that the case, that they occasionally occur sympatrically but don't form polyploids in Europe? And part of it might have to do with the ecology. So in general, they are allopatric, but when they are sympatric, they perhaps don't have the same, maybe they're separated by their pollinators. We actually don't know very much about the pollinators in the native range. What we know in the introduced range is that they are visited by a whole suite of similar visitors, so that there are many opportunities for pollen transfer in in the new world, in the
But that's easily testable. That's easy testable,
right,
with artificial pollination.
right.
Yeah.
So we know that there's an opportunity for pollination very easily in the U.S. But but that doesn't mean that there's going to be a polyploid, right? But what we think is actually potentially driving the polyploidy is the really unusual weather spurts that can happen in that area. So it's an area where, so it's the inland Northwest, it's dry. It's east of the desert, of the inland desert, but it's still quite dry. And it has a continental climate, but it's in an area that often gets like these Arctic blasts from Canada and this warm weather that comes up from through the Columbia River Valley. And so you end up with very sudden changes in temperature, where you can have a heat shock one day, and like the next day it could snow. And can happen in May and June, when the plants are just starting to flower. And we do know that one of the main triggers for polyploid gamete formation, or unreduced gamete formation, is temperature shock. Both heat and cold shock can lead to that. We've tried to get many to actually explore that, but we haven't been successful. But I think that that's also something that that could be looked at in more detail. And then, there's a lot of suggestion in the literature that sometimes hybridization itself can lead to meiotic breakdown and the formation of unreduced gametes. So if there is just a higher hybridization rate because of these shared visitors, inflorescence visitors, then there could be just more hybridization, which could lead to higher polyploidization. So we have a lot of hypotheses, but not a lot of data to explain the Tragopogon system. But I think more generally, it's a really important question.
And why is tomato not tetraploid then?
I don't know. I mean, there you've got close relatives. I mean, sometimes we think that chromosome size may actually be at play to determine whether a group might undergo polyploid formation. Because if the nucleus can only be so big without making the whole cell bigger, that there's that whole both nucleus and cell size dynamic. And so there are some hypotheses. I know, but it shouldn't be an issue between the two of those. So sorry, can't help you on that one.
Fabulous seminar. Thank you so much.
Oh, thank you.
How big is the Tragopogon genome?
The diploid is about 2.7 gigabases, so it's
okay, it's pretty big.
Bigger than we would like. Put it that way.
Yeah, I know.
It has a lot of repeats. It has a lot of repeats in it. So yeah.
So the tetraploid is 5 gigs, something like that.
Yeah, something like that. Yeah.
So you have a bazillion barley genomes to sequence, essentially. So
right. Yeah.
Okay. Are the polyploid, the Tragopogon polyploids, are they more vigorous in their growth? Are they bigger? Are they?
Yeah, they are a lot bigger, and that's been captured in a number of some of the classic literature on the Tragopogon system by Ownby and some of his students. We've done some greenhouse studies that have measured a lot of that, and we have a former undergrad, so long ago an undergrad that he went to grad school, and I saw him at PAG a year ago, and he already has a job. So so that's how long ago the data were collected and still not published, but we're working on it. But yeah, the tetraploids are a lot bigger, and they generally produce larger inflorescences. The inflorescences have more seeds. The seeds are larger, and probably I would say that they're, I don't know if we actually have all of the data on this, but I think that they're a bit more robust, and their germination percentages are higher.
Okay. Yeah, I was just kind of curious about have you ever so why are again, to Mark's question, why are these guys actually forming polyploids and things? Have you ever looked at like if their microbiome communities change when they go from the diploid to the polyploid? Has anybody done anything like that?
Yeah, no, that's a great question. I mean, when we were at Washington State, it was easier to think about doing ecology because they were literally in our yard, as non-native plants, we literally had them in our backyard. And so we did do a lot more like local ecology study there. Since we've been in Florida, which has now been almost 20 years, it's been a lot harder to do some of the field work. But, interestingly, the microbiome, has come up as a concept, subsequent to the time that we lived there. We have a graduate student who's working on a different polyploid system, and her work, it's an autopolyploid. It occurs in the Appalachian Mountains. And her work will include some microbiome studies of the diploids, triploids, and autotetraploids. But yeah, it would be really interesting to go back and do that with the Tragopogons too. One interesting thing we did find, and this was, one of those accidental observations from within a greenhouse study that we were doing at one point. The student who was working in the greenhouse, just doing the watering and stuff, was actually an ecologist, and he asked us one time. He said, there aren't any aphids on this one species. He said, is this one of the tetraploids? We're like, yeah. So he could tell that there was actually resistance to some of the greenhouse pests in the tetraploids that, and they seem to sort of get after the diploids. So I think that there are probably a lot of things associated with the chemistry and with the probably their whole microbiome that we don't know anything about. But yeah, really.
Okay. I'm going to just follow up with a question. Then we have another question from the chat room. I mean, so you had a kind of one of your concluding slides was, you have the polyploidy and then you formation, and then you maybe have one that just takes off. And dominates. Have you seen that in the Tragopogon study?
So in terms of, so almost every population is a different genotype. And so I think we're at an earlier phase there where we see more of the popping up of the different genotypes and that diversity. I don't think it's maybe far enough along that spectrum to have gotten to that point. Although, if we look at the complex in Spain, maybe we could comment on that a little bit more. So there are two tetraploid origins that occurred within the Pyrenees. Both have very narrow distributions. But the other one is a much broader distribution across sort of the north central part of Spain. So whether that's an older origin or whether it's, whether it's just been able to colonize better and expand its range better, we haven't investigated that. But that could maybe represent one of those cases of one that's, sort of taken off relative to the others.
Okay, well, I think I see a couple questions in the chat box. And can you pull that up, Pam?
Yes, I can. Yep. And so
okay, it looks like there's two questions.
Yeah. So the first one is from Pedro. Do you believe that polyploidy could be one of the answers to Darwin's abominable mystery about the sudden appearance and diversification of angiosperms in the Cretaceous? Well, that ties together two of my favorite topics, so polyploidy and phylogeny. So, so of course, I'd like to speculate on that, but we don't yes, I do think that there could be something to that, because we see not only the ancient whole genome duplication that is found in the common ancestor of all angiosperms, but there are also a lot of additional early events in the magnoliids and some of the other early clades. And so a lot of that diversification that was happening early on in the angiosperms was also being accompanied by ancient duplications. And so again, if we think about ancient duplications as generating diversity, and that diversity leads to morphological or physiological or other sorts of novelty, it could help explain some of those radiations. Doing this retrospectively, can we actually nail any of that down? I think that's pretty difficult, but it's certainly an interesting set of ways to think about how evolutionary changes involving whole genome duplication may have played out over some of these deeper timeframes. And it would be interesting to actually think about maybe modeling some of those things. Like if you have duplications that generate a certain number of genes, and if those genes happen to be involved in certain types of traits that we know are important, what might be the consequence of that? So yeah, thanks for that question. It's a really interesting question. And the next one is from Alice. Polyploidization can be advantageous in a changing environment, but on the other side, genomic redundancy can eventually reduce with genes that may be lost or subfunctionalized, which may be the successful compromise for polyploid evolution and spreading. Yeah, also really good questions. So I kind of think about, a lot of these things as, they're sort of along the lines of a continuum, and it may be that under certain circumstances, one outcome is favored, and under other circumstances, the alternative is favored. So, under certain circumstances, maintaining duplicate copies so that you have lots of gene product might be important. Under other circumstances, maybe that's too expensive. And so subfunctionalizing so that different copies take on different functions would be beneficial. And, I think there's a lot of opportunity for modeling what might happen. So under certain sorts of environmental limitations, what sorts of things might you see? Under other circumstances, what might you see? So I think that there's a lot of potential for exploring those sorts of questions through modeling, but also maybe through experimental evolution. So, people are starting to do work with duckweed and multiple species of duckweeds, for example, where with polyploids and diploid parents grown in vats, like people do, like with bacterial sorts of experiments. And it's possible to, set up environmental restrictions or limitations based on nutrients or salinity or some sorts of adding stresses and so on, and seeing how polyploids respond, or including synthetic polyploids that could be made and put into the system, but how the polyploids respond to the diploids and what sorts of changes actually are made after many hundreds of generations, because you can get them really fast. Not quite as fast as you can in a computer, but pretty fast. So yeah, good question. Okay, so the next question from Michael. There used to be the genomic shock hypothesis regarding epigenetic changes after allopolyploid formation. Do you have observations into that direction? Thanks for that question. We did make those some observations along those lines in a paper that Richard Buggs was the lead author of in, I think it was 2011, in Current Biology. And so, yeah, there seem to be some things that happen very quickly after polyploid formation at the genome structural level, and maybe at the transcriptomic level as well, that suggests that we could be witnessing some short periods of shock before things settle down. It's kind of hard to put all of this on a temporal scale, and we can look at generations in our synthetics and compare those results with what we observe in nature over the course of, at the most, 45 or so generations. But all of that is essentially instantaneous compared to evolutionary time. So, you know, looking at how long that period of shock might be in something that's existed for a longer period of time, I think is an interesting thing to think about. But yeah, but we do see some evidence of sort of rapid changes right after polyploidization. So, for example, in those two big charts that I showed of the homolog loss and the transcriptomic changes, the expression changes, the individuals on the right half, basically, of each of those charts were synthetics, and they showed essentially the same patterns as the natural populations, meaning that within, you know, one generation or so of being polyploid, they were already shutting down some of those genes in terms of their expression patterns, and they were already losing copies from the genome. And that's, I mean, that was described from wheat a really long time ago, 20 years ago, probably, that in the in F1 hybrids in wheat and early generation polyploids, they were already undergoing those sorts of changes. And so we do see that, like, within a few generations, the same patterns that we see in the natural populations have already originated. Okay, let's see, next question. I had a general question about what can occur in polyploid species. I'm working on a polyploid wild rice species, which appears to have one parental subgenome 2 times bigger than the other parental subgenome. I'm just generally curious if you've seen this in polyploid species. So none of the polyploids that we work on have that pattern, but that is an interesting one in itself. So if the subgenome that's twice as big is not itself polyploid, but is bigger just because of transposable elements or repeated sequences, it would be interesting to see then what happens to all of those sequences in the polyploid. Are some of those lost, or what happens? Or are they retained faithfully from that parent? But yeah, it sounds really interesting, but I don't have any experience with that, sorry. Oh, very interesting question from Mohammed. Can you share your thoughts on what you think are the essential ingredients of creating a high-performing scientific team? That is a really interesting and difficult question. I'm sure there's a lot of luck involved, you know, but I think what it tends to happen, and this is what we see in selection of, you know, graduate students and postdocs for our lab, that over time, you know, you develop a group of people who work really well together, and that sort of becomes true breeding in a way, that as you add the next graduate student to your group, or the next postdoc, if the people who are involved in the, you know, are currently present, contribute to the selection of the next students, for example, you tend to be able to retain the camaraderie and team atmosphere and family atmosphere that you had before. And I mean, and I don't mean this in a way that you end up only selecting people who are just like you. Hopefully, you know, we're doing a better job of also incorporating diversity across numerous spectra. But I think it's but sort of looking for those elements of teamwork and of collegiality and the willingness to work together as a team, and sort of selecting for that. You know, I think helps. But you know, how you assemble that to begin with, you know, maybe it's maybe that's just sort of luck. I'm not really sure. But we have been incredibly fortunate to have had just amazing students and postdocs working with us, and then, you know, lots of teams of collaborators from other institutions too. So yeah, I feel very fortunate. Okay, next question from Natalie. Do you think that an allopolyploid from species A and B would necessarily outperform the F1 hybrid from the same species? So that is also an interesting question. And I think it would come down to whether you're thinking about multi-generational performance or just that one growth season. So a lot of times F1s will be bigger, will have a lot of the same characteristics that the polyploid has. But in most cases, the F1 will be sterile. So if you're interested in being able to generate something that will continue to breed without having to recreate it every year, then it might, then the polyploid would be better. So if you just think of corn, I grew up in Iowa, so I know corn really well. I worked in cornfields while I was growing up. The corn that is the crop is, it's an F1 made from inbred lines, and it performs awesomely, but it's not true breeding. So, they're not sterile, but you're not going to get the same product when those individuals reproduce. So, so there could be a lot of genetic reasons why a polyploid would be superior either as a product.
Oh, lost her. Tragic.
No, that's our first time.
Yeah, crash out. Maybe the windmills are frozen in Florida as well.
I wonder if she had a timer on her. I wonder, did she time out? Do you think she? No, she. I don't think.
But we're the hosts.
Yeah.
What a pity.
Maybe her computer went out.
What a great talk. But Natalie's question, I think, was like, can you think of a polyploid as an immortalized F1 hybrid, with the heterotic advantage of a F1 hybrid, but immortalized, which I thought was a pretty good question.
Haven't had so many questions in the chat box. Everybody's scared to ask a question.
Yeah, but they're asking good questions by
they are.
Right?
But it'd be good to have a live question.
Well, yeah, yes.
Don't be afraid.
And she's fantastic, isn't she? There's another question from Lewis.
Yeah.
Prospecting. Do you think polyploids have an advantage?
Let me.
Yes,
everybody, hang on for a minute. I'm going to email Pam and see if she's something. Maybe her computer decided to.
Maybe the battery went flat. Yeah.
upgrade its operating system.
Yes. So while you're doing that, I should ask you, Rod, why is Ariza sativa not tetraploid? Why haven't they done tetraploid rice as part of breeding crop improvement for rice? Same questions for tomato.
I think they've been trying to. I think they've been trying to do that.
They've had 8,000 years to do it. Come on.
Yeah. Well, now we have the Alta that's has been neo-domesticated.
For sure. Yeah.
So,
hey, she's back.
Polyploid. Oh, you're back.
Fantastic. Muted, but back.
Oh, there. No, I tried to unmute it. It didn't work. Yeah, sorry about that. I got kicked off. Had to rejoin.
Gosh.
So I don't know where I got in my answer that you guys heard. I was talking away about corn, but you guys didn't hear any of it. I don't know. All of a sudden I noticed everything is frozen except me. So that's not a good sign.
No, it's not, is it? Which question was it you were hitting?
It was the one from Natalie, I think, right?
Oh, it was Natalie's one.
Yeah.
So, it was.
I don't know if I managed to, if you guys managed to hear an answer. It was a lovely answer, I'll tell you, but no, I'm just kidding. I'm just kidding.
So you've been talking to yourself for 5 minutes?
I was talking to myself for a few minutes, apparently, yeah.
We got halfway through, I think. But,
okay, so I think I probably hit it by the time I hit my point. By then, I probably just repeated it. But basically, I guess I was just thinking that if you're not worried about the reproductive aspect of it, an F1 and an allopolyploid would probably perform generally similarly in terms of
Yeah.
growth over the growing season. So,
It's sort of like an immortalized heterotic advantage.
right, exactly.
Yeah. We should probably let Pamela go, Rod.
Well, I think we have one final question, and
One more
then
question.
yeah, from Luis.
So I'm not, I think because I got dropped, I don't have any of the previous chat.
Okay, the
Okay.
last question is, thanks for the great talk. Do you think polyploids have an advantage over diploids?
Yeah, so also.
You missed a line there, Rod. Rod, you missed an important line.
All right.
From the bioprospecting perspective.
With context, bioprospecting, exactly.
Yeah. I think, I do think that there are some really interesting potential things that polyploids have that diploids don't. And part of that can be because a hybrid or a polyploid can actually generate novel chemical compounds relative to diploids. I think back to some of Don Levin's beautiful work in flavonoid chemistry from the 1970s. And they found lots of novel flavonoids in hybrids relative to the diploid parents. And I remember we used to see the same things doing allozyme analyses, things that are, let's say, that form dimers in, like, say, proteins that form dimers in diploids and that still form dimers in tetraploids, but when they're going to form their dimers, they can actually pull one subunit from one parental product and another from the other one, and you can make these heterodimeric proteins, for example, that are unique relative to anything that either of the diploids could produce. So I think if we're thinking about chemistry and various compounds that could be important in medicine or in industry, that hybrids and polyploids have, they're not the sum of their parental parts. They've got these additional novel compounds that could be useful.
Okay. Thank you, Pam. Yeah, a lot of chat questions and some great talk, good, excellent discussion. So thank you so much. And I think we're going to meet up after this for
I have a separate link to come back in to talk to that. Okay, great. And
so let's all applaud Dr. Soltis.
Thank you.